Hello everyone, welcome to my YouTube channel. Today we'll briefly talk about glycogen storage diseases. First of all, what is glycogen? What is glycogen storage diseases? By name, glycogen is stored. That is, it is not metabolized or broken down, which leads to glycogen accumulation within the cells, producing variety of symptoms, which we'll talk later. It has autosomal recessive type of inheritance, like various of the enzymopathies in the bodies, that is enzyme deficiencies in the metabolic pathway, they inherit by autosomal recessive inheritance. First of all, we'll talk about the glycogen. Glycogen is a polymer, that is storage form of glucose. It is stored in the muscle liver. Now see the structure. This is the structure of glycogen. Glycogen here. This is a glycogen chain. Here is a branch. This is an individual glucose molecule, right? This cyclic form of glucose. Now in this, we know glucose has 6 carbon. Let's count the glucose. First one is near the functional group, right? This is a functional group. 1, 2, 3, 4, 5, 6. So carbon 1 of one glucose molecule is combined with 4. 1, 2, 3, 4. 4 of the other. So alpha 1 4 glycosidic linkage. This connect one glucose molecule to the other in the glycogen. And here is the exception in case of branch. Here carbon 6 of one glucose molecule and carbon 1 of the other glucose molecule, forming the 1,6 bond is formed in the branch. So, glycogen is stored in the muscle and liver. These are the two places where, two organs where muscle and glycogen is stored. There is difference in glycogen of muscle and glycogen of liver. What is the difference? It is due to a single enzyme deficiency in muscle. Glucose 6-phosphatase enzyme is deficient in muscle. So when glycogen is broken down, glycogen gets converted to glucose 6-phosphate and then to the glucose. So glucose 6-phosphatase is absent, so it cannot be converted to glucose. Glucose 6-phosphate cannot get out of the cell, so glucose is not formed. It does not contribute to the muscle glycogen does not contribute to the blood glucose. But in liver, glycogen gets broken down to form the glucose and it gets liberated to the circulation, which contributes to the blood glucose during the fasting state. Now we can say muscle is a selfish and liver thinks for everybody, right? This is the difference between the glycogen in the muscle and glycogen in the liver. Now let's talk about the pathway in steady state. After you take a meal, after you eat glucose rich food, glucose is absorbed. There is increased glucose in the circulation in the blood, which gets converted to the glucose 6-phosphate. Normally glucose 6-phosphate goes into the glycolysis pathway and produces the energy in the form of ATP. But when there is excess glucose immediately after you fed, we are not supposed to use all the energy from the food within 30 minutes, right? So we are not going into glycolysis pathway. Rather, for the future use, when we have excess now, so that we can use in future, we convert this glucose 6-phosphate. That glucose 6-phosphate get diverted to the glycogen synthesis pathway. Right? This is a glycogen synthesis pathway. How the glycogen is formed. First of all, glucose 6-phosphate to the glucose 1-phosphate, 6-phosphate and 1-phosphate. There is a difference in the position of the phosphate. So phosphoglucomutase, only the configuration is changed. They are just the same molecules. So configuration is changed. So phosphoglucomutase is an enzyme. Now when glucose 1-phosphate is formed, it gets combined with UDP with the help of triphosphate with the help of UDP glucose pyrophosphorylase to form UDP glucose. Already have one phosphate at 1, then another phosphate is added, so diphosphate. From here, two phosphate are left, forming pyrophosphate. Now, when UDP glucose is formed with the help of enzyme glycogen synthase, which is a key enzyme in glycogen synthesis pathway, it gets incorporated in a glycogen chain. Here is a glycogen chain, right? It gets incorporated here. New glucose molecule gets incorporated here. Then again and again and again. In this way, glycogen chain is lengthened. The glycogen chain is formed. This is how glycogen is synthesized. This is known as glycogenesis pathway. Now, in fasting state, when you have nothing to eat, or when you are in fast, is provided with the help of glucose, which comes from the glycogen. Glycogen is broken down. The pathway known as glycogenolysis. Right? This is a lysis pathway. Broken down. Glycogen is broken down to release the glucose. In presence of enzyme glucagon, which comes during the fasting state. Right? It increases the activity of glycogen phosphorylase. Glucagon increases the activity of glycogen phosphorylase, which is a key enzyme in the glycogenolysis pathway. Now, by the name, let's talk about glycogen phosphorylase enzyme. By the name, it phosphorylates the glucose in the glycogen chain. So when it phosphorylates the glucose, that is phosphate is added, forming the phosphate bond, and phosphate is always a high energy bond, which is high energy means unstable. So glucose along with added phosphate is liberated. So here comes the name phosphorylase. You can remember like this. Now glucose polymer, which is glycogen, right? Here this way, glycogen phosphorylase at the phosphate B6 coenzyme is required. So glucose 1 phosphate is liberated by the glycogen phosphorylase. Now when glucose 1 phosphate is formed, it gets converted to the glucose 6 phosphate by the same enzyme, which is reversible reaction glucose phosphoglucomutase. Glucose 6 phosphate is formed. With the help of glucose 6 phosphatase converted to glucose in liver, which is liberated to the circulation, but in muscle it is absent. So formed glucose 6 phosphate converted to the glycolysis pathway within the muscle itself for its own use. Now this is how glycogen is broken down. First of all, glycogen phosphorylase liberate the glucose. It comes how the branch is broken down. How is branch broken? The answer is by the same phosphorylase enzyme till 3 to 4 carbon is till 3 to 4 glucose molecule is left in the branch, right? Like this way. These are broken down till 3 to 4 glycogen are broken down. Now in this condition, glycogen phosphorylase cannot function. The enzyme comes known as debranching enzyme. This debranching enzyme, it now breaks this limit dextrin, which is known as what is limit dextrin? Limit dextrin is a glycogen with 3 to 4 glucose, only 3 to 4 glucose molecules in branch, right? Now this is broken down by limit dextrin, liberating the glucose. This is how glycogen is broken down. After the work of debranching enzyme, this unbranched chain is formed, right? This unbranched chain is formed. This unbranched chain is acted upon by alpha 1,4 glucosidase enzyme. This is enzyme present in lysosome, right? And in this way, directly by surpassing all these pathways, it directly forms the glucose. Alpha 1,4 glucosidase form the glucose. In this way, glycogen is broken down. Now let's see. As I have already told, Glycogen storage disease is a disorder of the glycogen breakdown or metabolism. So, what are the enzymes of glycogen metabolism which will be deficient? First of all, most important enzyme is glycogen phosphorylase. 1. Next is debranching enzyme. 2. Debranching enzyme. 2. Phosphorylase debranching enzyme. Third is glucose 6 phosphatase. After it gets converted to glucose 6 phosphate. Third is glucose 6 phosphatase. Fourth is alpha 1 for glucosidase. All these enzymes are important. Now, based on their deficiencies, these enzyme deficiencies, what are the enzymes we have talked? Phosphorylase, debranching enzyme, 6-phosphatase, alpha 1 for glucosidase. Right? As for these enzyme deficiencies, we have different disorders. Different types of glycogen storage diseases. Type 1, 2, 3 and 5. 4 we'll talk later. 4 is not that important. Type 1. We have mnemonics here. Very poor carbohydrate metabolism. Very poor carbohydrate metabolism. V. Von Zierk disease. Poor Pommes disease. C. Cory's disease. M. McArdle's disease. McArdle's disease. These are 4 glycogen storage diseases. 1, 2, 4, 1, 2, 3, 5. Now, how will you remember the enzyme deficiencies? G for von Zierk disease. From G of von Zierk disease, we'll remember glucose 6 phosphate is deficient in von Zierk disease. Now in Pommes disease, mnemonics is P. Pommes, P-O-M-P. 1 and 4 position there is P, right? So alpha 1 and 4 glucosidase is deficient in Pommes disease. C for Cory's, then D, debranching enzyme, CD, alphabetical order. Now McArdle's disease, M for McArdle's, M for Moser glycogen phosphorylase is deficient. Now 4 and 6 are remaining. 4 is Anderson's disease. We have talked 3 is AB, right? 3 is CD. 4 is AB, Anderson disease branching enzyme. And 6 is horse disease. S for horse, S for hepatic glycogen phosphorylase. This is about the pathway, right? First of all, this is the summary. First of all is CD. 4 is AB, Anderson disease branching enzyme. And 6 is horse disease. S for horse, S for hepatic glycogen phosphorylase. This is about the pathway, right? First of all, this is the summary. First of all, during the fasting stage, fed state, glucose comes into the circulation, converted to the glucose 6 phosphate. When there is excess glucose 6 phosphate, it gets diverted away from the glycolysis for the future use. First enzyme acted is phosphoglucomutase, forming glucose 1 phosphate. Then combined with the UTP, forming UDP glucose, the molecule which is attached to the glycogen chain with the help of glycogen synthase enzyme. And this is how it is synthesized. And after a chain is formed, then branching also occurs by branching enzyme. How a complete glycogen molecule is formed. After a complete glycogen molecule is formed, it is broken down by the pathway known as glycogenolysis. Now, the key enzyme is glycogen phosphorylase. Other enzyme is debranching enzyme, converted to the glucose 1 phosphate, then again to the 6 phosphate, the glucose 6 phosphatase. And after an unbranched form is formed, unbranched glycogen is formed, it is directly acted upon by alpha 1 4 glucosidase. These all together form the glucose. This is how glucose is formed from the glycogenolysis. Now